Hey, I'm Dr. Kyle Gears, and this is an episode of Thursday Brain Bytes, where we go over interesting neuroscience facts and how NeuroGlider can help you insert your electrodes for in vivo electrophysiology better. So for episode 1, we're going to go over some basic fundamentals of neuroscience and the underlying biological principles that underlie the electrophysiological action potential that you're recording during electrophysiology. So this is your brain. Well, this isn't your brain. This is a blue stuffed model of your brain, but they don't let me have brains on camera, so this is going to work. The brain itself is made of many different cell types, but the one we'll focus on today are excitatory neurons. And so a neuron is made of 3 specific cellular compartments: the dendritic field, the cell body, and the axon. So within a cell for neurotransmission, the dendritic compartment receives signals from axons from projecting neurons, and the axon is what sends signals from a neuron to its downstream neurons. So that junction between the axons from one neuron and the dendrites from another neuron is called the synapse. At a synaptic junction, you have synaptic vesicles in the axon which release neurotransmitters into the synaptic cleft. And these neurotransmitters diffuse across the synaptic cleft to ligand-gated ion channels of the dendrite. If we look at this up close, what you have is a lot of positively charged ions outside of the cell in the extracellular space. You then have a mix of ligand-gated ion channels, denoted here in blue, and voltage-gated ion channels in green. So neuromessengers for excitatory synapses, these would be glutamate, diffuse across the synaptic cleft and bind to ligand-gated ion channels. When the neuromessenger binds, positive ions flow down the concentration gradient into the neuron. So in the extracellular space, you actually have in the cerebrospinal fluid an excess concentration of sodium and calcium. When neuromessengers bind, the ion channel opens, and this sodium and calcium is free to flow into the intracellular space, causing a flux of current. So when you have this change in membrane potential, voltage-gated ion channels then open, allowing further influx of cations, further increasing the membrane potential of the neuron. When we have electrodes placed in the extracellular space, we're measuring this flux of cations from the extracellular space to the intracellular space. And that's your Thursday Brain Bite.